Hi, my name is Engelhart. I'm an assistant professor at the University of Minnesota in the Department of Genetics, Cell Biology, and Development. And I'm going to tell you about some work that's been done and is ongoing on the search for life on Mars. So Mars has historically and still is a really attractive place for potentially finding life on another extraterrestrial body because there's a lot of similarities to Earth. And so it looks like a place we might be able to find life that's similar to that exists on Earth using the same types of biochemistry that you learned about. Um, Based on what we know on Mars, it's unlikely we're going to find, macroscopic life. We're obviously not looking for little green men. So most of what we're looking for is basically microorganisms, uh, things that are able to do metabolism, sort of with the inventory of small molecules that we have on Mars. Some of the early work that goes into this is from the Viking missions in the 1970s. And there was sort of a suite of experiments for biology that the Viking lander did in Viking landers did in 1976. And one involved taking a gas chromatograph and interface to a mass spectrometer and heating up the soil on Mars and looking for organics, with the idea being that life is making organics. And if we find some, there might be life there. And this one, what they found was that they didn't really find organics. What they found was chlorinated hydrocarbons. So if you work in a chemistry lab, you may have used dichloromethane. That was one of the ones that they found. At the time, they attributed this to likely coming from cleaning fluid that they had used in preparing the components of the lander. This has since been reconsidered. We'll get back to that a little bit later, but this one at the time didn't look so good. There's another experiment that they did called the gas exchange experiment. And the idea there was they wanted to look if basically a sample from Mars would do things like consume oxygen or produce CO2. Um, some radioactivity was released here, but again, it was difficult and there's still disagreement about whether these came out of biological activity or sort of abiotic processes. And this is something that comes up really routinely in these experiments looking for life on other celestial bodies. And that's that it's difficult to discern whether something is happening due to life or some abiotic process. Since then, we've learned a lot about these experiments and perhaps reinterpreted them in some ways, and that's kind of guided the search for life on Mars and other bodies since then. One strategy that NASA has adopted and people largely think about is called follow the water, with the idea being that life as we know it, you and I are about 2/3 water, most life is. If we look for places that water was likely to exist, that's probably somewhere that has conditions that are hospitable in some ways for life to form. There are features on Mars we know that look like this. The surface of Mars kind of bears hallmarks of the previous existence of liquid water. There are things that look like erosion, so basically dried up rivers and lakes and things like that. So most of what I've told you so far, again, goes back to the mid 1970s. In the mid 2000s, there was some work from the Phoenix lander that was later confirmed by two other rovers, Spirit and Opportunity, that again kind of caused us to reinterpret some of the results from the Viking missions. And so one big part of the story was that they found a lot of a chemical called perchlorate looked to be present on the surface of Mars. This was a complete surprise. They really weren't looking for it. What they had done at the time was sent a probe that was looking for another ion called nitrate in the soil. And turns out the way they were detecting nitrate could detect perchlorate as well. And this was actually the only explanation for it because the signal that they got from that probe that was looking for nitrate gave such a high signal that it really couldn't be explained by nitrate. The mass of the soil would have been more than the mass of apparent nitrate. So based on some other experiments that they did alongside with it, they basically assigned this as perchlorate and these other oxychlorine species that are present there. And so this has really shaped the way that we're looking at what the geochemical environment on Mars looks like in terms of the search for life. And so perchlorate is, kind of a good news, bad news story. It's so the bad news, first of all, is that it's toxic. Uh, you and I, if we were to ingest perchlorate, it's toxic to our thyroid. Um, it is a pretty strong oxidizer. And so, thinking about how life might use it, there's actually organisms on Earth that can basically breathe perchlorate a lot like they breathe oxygen. People have also talked about it beyond the search for life as a possible, natural resource on Mars, for perhaps terraforming it. If you've ever used, well, hopefully you haven't used an oxygen generator on a plane, but if you've been through the safety instructions on a flight, when they talk about the oxygen mask dropping down, that oxygen generator that's connected to the mask actually uses a compound that's closely related to perchlorate called chlorate, that when you heat it up, will decompose and make oxygen. And so this is a potential source for, not just fuel, but also, breathable oxygen. This also is usable as a fuel. The space shuttle, some other rockets have used perchlorate, actually as the oxidizer that's present in their rockets. And then finally, and perhaps most interestingly to what we're looking at here, is that perchlorate uh, has a really high affinity for water. If you take a sample of pure magnesium perchlorate, so if you were to buy from Sigma Aldrich or any chemical company magnesium perchlorate, it's a white solid. It doesn't really look like much, but pretty much any other salt or a lot of other salts. If you were to take that and leave it out on a countertop or a bench top in the lab, what you'd find is if you came back a few hours later, especially on, kind of a humid summer day, there wouldn't be that nice white powder there anymore. There'd be a puddle there. And that's because perchlorate is very hygroscopic. It has a really high affinity for water. And so thinking about life as, the one matrix that we know, or sorry, water is the one matrix that we know for life. The fact that perchlorate has such a high affinity for water has kind of shaped the way that we're thinking about, especially near surface water on Mars. And there's a number of groups that have presented evidence that perchlorate or oxychlorine species that are present on the Martian surface could be absorbing and releasing periodically water and actually making liquid water present on the surface. If this is present right now, it is going to be obviously in the presence of that perchlorate, which is going to be pretty salty. These are going to be brines. That puts some constraints on what life could exist in sort of those near surface environments, because the different organisms have different tolerances for salt. And the concentrations that we're talking about get pretty high. This is multiple molar salt basically present in these brines. There are some terran organisms that we know on Earth that can survive in the presence of these really high concentrations of perchlorate. And so it is possible that, these brines actually could support life. Another downside that this comes with, though, is that perchlorate actually enhances the basically sterilizing capacity of ultraviolet light. And there's a lot of ultraviolet light that reaches the Martian surface. So that perchlorate kind of adds something to that, and actually makes that UV more sterilizing. And so this is another reason that some people that are looking at life on Mars have speculated that the subsurface could be the best place to look for this. Once you get very far below the surface at all, you kind of solve that UV problem, because just like, if something's buried under a bit of dirt, it's protected from sunlight. The same is true of something on Mars. If you have even just a few millimeters of regolith there, so in the near surface or just barely subsurface, a lot of that UV problem kind of gets mitigated there. And then, of course, as we get further down in the surface, or the subsurface, that opens up new opportunities for looking for, potentially fresh water and other interesting geological formations. And so this past year was a really exciting time for the search for life on Mars. Mars 2020, the NASA mission launched. Europe and China also launched rovers there. And this is going to be basically setting the stage for future sample return missions from Mars. And the reason that sample return is so important and could potentially be so powerful in telling us about what's present on Mars or the search for life on Mars is because that's going to enable us to do experiments that we really haven't been able to do before. If you, sort of think about how we're getting to Mars right now with these spacecraft that we've been talking about, this occurs by basically using the orbit and the gravity of Mars and Earth, this multi-month journey to get whatever craft you're sending there. And as a result, the only experiments that we're able to do there are essentially limited by what we can basically put on one of these spacecrafts that's compatible with that type of journey. And everything that I've told you so far about these missions that have given, in some cases promising, but perhaps inconclusive results, a lot of the reasons that they are inconclusive are that we can't do the type of follow-up experiments that we're able to do in a lab on Earth where we have all the kind of fancy instrumentation that we have there. And so right now, a lot of what's done uses basically simulants of Martian regolith or soil. And this is one such example that's been generated based on some studies that have been done in some of these missions that sort of allows us to do some of these experiments in the laboratory based on what we know about Martian geochemistry. But of course, nothing beats having these actual samples now. And so what we're doing right now is drilling into the surface and subsurface of Mars to get samples that are currently being cached with the idea that these are eventually going to be returned to Earth. And the result of that's going to be that we're able to interrogate them in ways that are a lot more sophisticated than what we're able to put on these spacecraft that, we're basically limited by what's going to be able to make this, sort of double gravity trick, long journey from Earth to Mars. So it's a really exciting time for the search for life on Mars, precisely because of this. Because in the coming decade or two, we're going to learn a lot about this. We're really setting the stage for this, and it's a very exciting time to be searching for life on Mars. Thank you.